Thanks everyone for coming to another session of the Aspirational Neuroscience Journal Clubs. We're going to be looking at this paper today, the circuit reorganization in the Drosophila mushroom body calyx accompanies memory consolidation by Baltruschat. I'm not sure I'm pronouncing that right. Baltruschat and out of the Tavosanis lab in Germany. I think it's a fun paper. But before we get through the paper, just to give people a bit of context on what we're doing here. So ultimately, the aspirational neuroscience community is interested in this memory decoding challenge, which is this demonstration of the decoding of a non-trivial memory from the static map of synaptic connectivity. So rather than taking an active, freely moving animal, looking at calcium dynamics or things like that in its brain and trying to decode memories that way, this is instead just taking a static connectome and trying to see if we can pull anything out of that. So canonically, we've been looking at things like, would you be able to pull the song from a zebrafinch brain, the unique song that it sang, out of that? Or recently, we've been looking at things like grid cell properties, or visual learning, or visual environmental properties from the visual cortex of an animal. And we've been doing this because at the moment, the current journal club series we're focusing on is looking at examples of model systems and experiments that might actually be capable of winning the memory decoding challenge. We had a whole bunch of papers, particularly last year, where we were looking at candidates for the annual research awards, which are these awards we give out, which are more interim sort of prizes for things that don't quite win the entire memory decoding prize, but are a step along the way to getting there. But at the moment, we're sort of taking a step back to look at model organisms, model systems that might give us an idea of what a winning experiment would look like for the overall memory decoding prize. Although I think today's paper sort of is informative about both aspects. So as I was saying before I started the journal club proper, my background is in rodent behavioral neuroscience rather than Drosophila. So when I was getting into this paper, I had to learn quite a few of the basics of how Drosophila neuroscience and Drosophila behavioral neuroscience in particular actually works. So in case some of you are similarly naive to the ways of Drosophila, I will run you through this briefly. So this is Drosophila, a fruit fly. And the way that behavioral neuroscience is typically done in these animals is the same sort of operant conditioning of
Looks like Ariel froze there. We'll just wait for him to pop back in.
So while we're waiting, Ken, I've always wanted to ask, what is all of that behind you? It's like real mad scientist type stuff.
Hello.
Multi-sim.
Hi, Ariel. Great to have you back.
Yeah, I was in the middle of presenting and I was told I'd lost my network connection, so I've switched over to mobile. Thankfully, this exists these days, so I cannot be prevented from presenting this paper, much as my internet might try at me. We are back.
And you were just trying to explain operant conditioning on fruit fly.
Yeah, is this... Can you see? It's not really... why isn't this working? Sorry. Let me just try and get this to work again. There we go. I think that should be working. Okay, operant conditioning in fruit flies. I was comparing it to rodent operant conditioning, but rather than having a big, box that you put them in with a grill to shock them and different scents, use these clear plastic tubes where you can put them in a tube. You can put the little odor into the tube, and then you can either have a shock tube, for example, where you pair an odor with these little strips, or you can have a testing sort of setup here, where you have two different odors on two different sides, and you just see how many flies go in one direction versus flies go in the other direction. And you can do the same sort of thing, but with appetitive stimuli rather than shock stimuli as well, as we'll get to in a minute. Just to check, when I move my mouse around, can you guys see where my mouse position is?
Yeah,
someone give me a thumb. Yeah.
It's pretty small, but it's visible.
Great. So that's the basics of operant learning. And typically in this study and in many studies with Drosophila, the way they present their conditioned stimulus is an odor. So they do odor pairing. And to run you briefly through the anatomy of how Drosophila detect odors, they have these two antennae. There are olfactory neurons in these antennae that project into these antennal lobes, which take in the information from the antenna. And it's in these mushroom bodies where the magical learning, the pairing of these conditioned stimuli and these unconditioned stimuli is thought to happen. To give you a more sort of physiological view, here they are here at the dorsal portion of this Drosophila brain. And to run you through the specific physiology that we're going to be looking at a lot in these paper, the focus is on how these olfactory receptor neurons that are in the antenna send their axons into the antennal lobe, where they meet up with these projection neurons. And it's the projection neurons that run from the antennal lobe into the mushroom body, where they meet these Kenyan cells. This is what we're going to be focusing on for the purpose of this paper. And it's in the mushroom body where a large degree of focus on how Drosophila learning and memory has been happening. Because in part, people think that the mushroom body is somewhat analogous to the human or mammalian cerebellum. Randall, yes.
Yeah, sorry. This is kind of where I guess I'm going to throw in my question about the circuit that I sent you an email about earlier, because I was getting a little confused. The paper doesn't really give us a nice diagram. Instead, it has a description of which cells are connecting to what cells. And there's 33 excitatory cholinergic contacts, 270 postsynaptic profiles, each KC contact boutons with one claw. But then comes this thing that I was a little bit surprised about. So then it said within MG, bouton received presynaptic input from 4 cells, and those were then from Kenyan cells. So I was getting the impression that there's some kind of a loop in there that I didn't see in most of the diagrams. Do you know if it's recursive?
I haven't seen. Hold on. Let's get onto the next slide where I've got a slightly stronger diagram. I have not. I've not heard of it being recursive in any of the reviews I've looked at so far. So the idea is, if we look at this,
I didn't see that on diagrams that I found either. That's why the paper threw me a little bit. But maybe I was misreading it.
Perhaps let me just run through again. So for everyone else, typically the model of how smell detection and learning works in Drosophila is you have on the antenna you have these olfactory receptor neurons that are sensitive to a particular kind of odor molecule. These then all project into all of the same olfactory receptor neuron type, so that have the same olfactory receptor, project into the same the same glomerulus in the antenna lobe. So each of these little lobes here, for example, takes in olfactory receptor neurons of the same smell type, basically, in a sense. And then each of those antenna lobe glomeruli sends projection neurons rather into the mushroom body, where it makes contacts with these Kenyan cells. And there's a limited number of projection neurons, but there's a lot more Kenyan cells. And each of these Kenyan cells is taking in inputs from multiple projection neurons. And then the Kenyan cells send these long axons throughout the mushroom body itself, where they make multiple contacts with these different kinds of mushroom body output neurons. And that's where traditionally learning has been thought to be occurring within the mushroom body. So if you look at a stylized diagram here on the right, we look at the differences where flies are learning to either avoid a stimulus or approach a stimulus. And what we think is happening historically is that when a particular smell comes into the olfactory receptor neurons, which then leads to activation in the projection neurons, which leads to a certain subset of Kenyan cells being activated, that this information about the smell, which is transmitted by these Kenyan cells, also receives inputs from these, let's say, dopaminergic neurons that tags it with a certain valence. Whether that's this is good, that's a good smell, we should go towards it, or that's bad, it's a painful stimulus, we should be avoiding it. And it's through this modulation of the Kenyan cell contacts onto the mushroom body output neuron contacts that the flies learn to avoid or approach particular smells. And this happens within all these different lobes within the mushroom body proper. I am not aware, Randall, of where recursion is supposed to come into this.
No, that's okay. This could just be because the way it was written in the paper was confusing to me, and then I couldn't find any diagram that specifically followed the way the paper was lining it up. I'm very happy to just go with a purely feed-forward system.
The only part of recursion I can think of is there is inhibitory modulation, where I think there's something like all of the Kenyan cells are contacted by this, and if I'm remembering this correctly, this one particularly large inhibitory neuron on each side. And it means that you get a winner takes most dynamics where you can't have too many of the Kenyan cells firing at once. It enforces a sort of sparse firing system. And that may involve Kenyan cell firing, activating the inhibitory neuron, which then inhibits the other Kenyan cells, which I guess is a kind of recursion, but it's a pretty limited form of recursion. But if there are any Drosophila experts who want to pipe up at any point, literally just pipe up and interrupt me, because you probably know better than I do as we go. So this is the canonical sort of learning circuitry that's examined in Drosophila papers, but it is not the learning circuitry that we're going to be looking at today, because despite almost all papers focusing on this side of things, what's happening within the mushroom body proper, and how it is that dopaminergic neurons are modulating the outputs of these mushroom body output neurons. Instead, today, we're going to focus on where the projection neurons actually contact these Kenyan cells in a region called the calyx, which you can see here is to the dorsolateral side of the mushroom body proper. Because it turns out in this paper, they're going to examine how there are some structural changes associated with learning that happen upstream of all this fancy dopaminergic neuron side within the calyx section itself. So yeah, again, this is the paper we're looking at, how there's circuit reorganization in the calyx that accompanies memory consolidation in Drosophila during olfactory learning. So in this particular paper, they focus on these DA1 projection neurons and the context they make within the calyx of the mushroom body. So in particular, in this paper, they end up using two different odorants, CVA and GA. And CVA is a smell, it's sort of a male-generated odor. So male Drosophila give off this particular odorant. And apparently for humans, it has a kind of fruity smell to it. And the reason why they focus in particular on DA1 is because the projection neurons from glomerulus DA1 in the antennal lobe are pretty specific to DA1, to responding to something like CVA. So they think they have a very neat population of projection neurons, which are quite specific to this kind of odor, where they're able to know that if they suspect going into the paper that if they see neurons that are responding to the CVA smell, that they're going to be from this DA1 glomerulus in the antennal lobe, that it's neatly labeled for them going forwards. And the other smell they use as a control throughout the paper is GA, which is just another smell. It has a floral smell to it. But specifically, they think that the olfactory receptor neurons that are sensitive to GA don't originate from this DA1 glomerulus. They originate from a different population of olfactory receptor neurons and then projection neurons. This is critical for what they're going to be exploring throughout the rest of the paper, that the projection neurons they're looking at, these DA1 projection neurons, are specific to CVA as opposed to GA. So that's the starting point. They're using these two smells. They're looking at projection neurons, these DA1 projection neurons, and they're looking at where these DA1 projection neurons enter into the calyx. So the point where they enter into the mushroom body and make contact with these Kenyan cells that are known from previous studies to be involved in learning and memory in the fly. The first thing they do is they establish that these DA1 projection neurons really are involved in detecting the CVA smell. So they have flies that have a calcium indicator on these Kenyan cells. So on the postsynaptic side of the projection neurons, they're monitoring calcium activity in response to presenting odors to the flies. And they label the DA1 projection neurons with TD tomato. So basically the axon terminals of the DA1 projection neurons are going to glow in red, and they look at the overlap between these within the calyx of the mushroom body, and they see that when they present different smells, particularly when they present CVA, that the overlap between the red, the DA1 projection neurons, and the activity they see is 85% or 84%. So basically, almost all the DA1 projection neurons that they're seeing are responding only to CVA, as opposed to other cells in the calyx which are responding to the solvents that they place the CVA into. So they use an ethanol water mix as their solvent that they're presenting CVA in. So this is just to say that indeed it seems to be the case that these DA1 projection neurons are responding quite specifically to CVA. Is everyone still with me before I continue on? Getting some thumbs ups? Good. All right, let's continue. So I'm just going to leave this up here to give you a bit of a sense of the circuitry as we continue. What they've known from previous studies is that each of these olfactory projection neurons, so these projection neurons carrying information about particular odors, when it arrives in the mushroom body calyx and it makes connections with these Kenyan cells, you have essentially the axon of the olfactory neuron, of the projection neuron terminates into a bouton, and then this bouton gets enwrapped by the dendrites of the Kenyan cells. about 11 different Kenyan cells will try and wrap itself around this single bouton of these projection neurons. And this has been termed a microglomerulus. So not a glomerulus like the big lobes that you see within the antennal lobe. These are little glomeruli, microglomeruli, that exist within the mushroom body calyx. And what they do in this study is they actually go back to a different connectome, one obtained from the Zheng et al. 2018 paper. And within that paper and their data set, so not data that was collected directly for this paper, they pick out a DA1 projection neuron. I don't know, they don't make clear how they chose the specific DA1 projection neuron within the paper, but they pick out a DA1 projection neuron. And what they did is they traced that neuron from the antennal lobe into the mushroom body calyx. And then they looked at all the different connections it made to other Kenyan cells. So it was showing that this single DA1 projection neuron is making this connection to a whole bunch of different Kenyan cells. So it's got like 8 of these gamma Kenyan cells, 3 of these AB, and you can see a set of these other ones. Randall?
Sorry to interrupt. It's when I look at that traced out DA1 PN neuron, I see that, yes, it's got that one branch that goes off to the bouton that they're now focusing on. And then I see it continues, and there's a set of other branches. It seems like there's much more going on down there. And I guess maybe I didn't pick this up in the paper, but why are they focusing on this particular bouton? Do we expect that there are further microglomeruli at each of those other boutons at the end of that axon, or what are those doing there? Any idea?
So yeah, so the projection neurons make 2 large groups of contacts. They go into the mushroom body, where they're thought to provide contacts that are involved in learning and memory. They also continue on into the lateral horn, which is a different body within the Drosophila, where they're thought to be involved in more sort of instinctual behaviors that don't have learning and memory associated with them. Now, again, I'm not a Drosophila researcher. I have not looked at what happens within the lateral horn, and maybe some learning happens there. But in the reviews I looked at, that was the distinction. It's like lateral horn for instinctual behaviors, mushroom body for actually learned behaviors.
Awesome. Thank you.
Yeah. So that is sort of the connectome aspect, where you can see the single DA1 projection neuron is making contacts with all these different Kenyan cells. And on the right here, they provide this nice little visualization of what these microglomeruli actually look like under electron microscope. So you can see that this bouton is really being enwrapped in the dendrites of all these different Kenyan cells. And on the left here, you can see this sort of interesting structure where this one active zone, this one sort of pre-synapse from this projection neuron is making simultaneous contact with all these different dendrites. This polyadic synapse is how they refer to it, which, again, I'm not a neuroanatomist or a connectomist, but is a feature that is not very common, as far as I'm aware, in mammalian brains. So it was kind of cool to see these differences here in the insect brain. They also critically, when they've done this investigation, they are able to do some actual quantification of the sorts of things they see here in this structural data. So they see that this DA1 projection neuron makes 33 excitatory cholinergic contacts that are all polyadic. Polyadic being has those one bouton to multiple dendrites at once. And it's opposed to 277 postsynaptic profiles. Then they go through all the details. And they say each Kenyan cell contacted the bouton with a single claw, which receives something like 8 to 25 presynaptic inputs from the projection neuron bouton, which is in line with previous estimates. So this is just quantifying the number of contacts, which they're doing with the electron microscopy. Now, I think that's just relevant to keep in mind when we're thinking about the numbers involved in these sorts of contacts. but we will come back to these details as we go through the rest of the paper. So that was taking data from the Zheng et al. Connectome that was done prior to this particular study, but they also do some structural work within this study itself. But all the work they do within this study is using confocal microscopy rather than electron microscopy. So what they want to establish here, now that they've identified their DA1 projection neurons, they know that they're CVA sensitive, and they want to be able to look at the contacts between their DA1 projection neurons and the Kenyan cells that they synapse onto. What they do in order to enable them to do that going forwards is that they label the DA1 projection neurons, their terminus, their active zones, with this marker BRP short cherry, which basically turns them red, as you can see here on the left. And they mark the postsynaptic densities of the Kenyan cells with this GFP tag on their acetylcholine receptors. So the postsynaptic densities are in green, the presynaptic contacts are in red, and they use that to look at where these DA1 projection neurons are contacting onto these Kenyan cells. And using a bit of computer assistance, they're able to quantify the number of DA1 contacts onto these Kenyan cells, and they use that to do a quantification of the number of microglomeruli that are occurring within the mushroom body calyx. So they're basically saying, of all these microglomeruli that are conveying information about all sorts of different odors, how many of them are conveying information about the odorant CVA, which is what they're interested in this paper. And critically, using this confocal microscopy setup, they're going to look at how do these microglomeruli change before and after appetitive learning occurs within Drosophila. So how do they actually do those learning and memory studies? So again, this is the sort of operant conditioning setup that they use with their Drosophila. So they have two phases. In the training phase, they pair one of the particular odorants with sugar, which essentially just involves putting the flies in a tube with sugar added to a little patch with one of the smells and not the other one. So in the CVA is the conditioned stimulus, the sugar is with the CVA. In the control GA with the conditioned stimulus, GA is with the sugar. And they also have this mock control where the two smells are present, but neither of them are paired with the sugar. So we've got CVA positive stimulus, GA positive stimulus, and mock. And these are the three types they're going to be using throughout the rest of the study. So that's the training phase, which takes place only over a couple of minutes. And then they have a testing phase, which is either almost immediately after the initial training or 24 hours after the initial training, where they place the flies in between two tubes, one filled with CVA, one filled with GA, and then they just test what proportion of flies go into the CVA versus what proportion of flies go into the GA as a sort of test of how well the flies have learned to associate sugar with one of these different smells. So we can see when they examine the short-term memory performance of flies that are trained in this way, that flies are definitely able to develop a preference for one of these conditioned stimulus. So if they were trained on, in blue, the GA, then they go for the GA. If they were trained in red on the CVA, they go for the CVA. And in mock, they have no particular preference for either the CVA or the GA. But they don't see any structural changes when they're looking at this 1 minute post-training in the Drosophila brain. So that means when they take the flies that have undergone this short-term memory training and immediately after the testing phase, they dissect out their brains and they perform this confocal microscopy, they're not seeing any sort of structural changes that have happened that quickly within the mushroom body calyx, which sort of makes sense. You'd expect that to be more immediate protein mediated or electrophysiologically mediated. That is in contrast to when you do long-term memory. So 24 hours later, the flies that have been trained to like CVA or to like GA are still showing this quite robust preference for whichever stimulus was paired with the sugar. But this time, you start to see some changes in the structures of the mushroom body calyx. So specifically, in flies that have been trained on CVA, you can see a reduction in the relative size of the microglomeruli. You can see a reduction in the relative lumen volume of the microglomeruli. And you can see an increase in the number of the microglomeruli in particular. And this, interestingly, is happening specifically for the DA1 microglomeruli in flies that have been trained on CVA, but it's not happening in the DA1 microglomeruli for flies that have been trained on GA. Now, that's good because it's a form of control where it's specific to the microglomeruli that are associated with projection neurons that detect CVA. It may well be the case that if you look in these flies that have been trained on GA as their condition stimulus, that there's another set of microglomeruli where you're seeing this increase in number and this decrease in size, but those aren't the ones that we're looking at here. We're looking specifically at these ones that are associated with the CVA. So what they're just showing is that this DA1 microglomeruli number increase is specific to flies that have actually been trained to have this positive association with CVA. So that's good, and it's a cool sort of structural change that's happening that's associated with this this long-term memory formation. That's kind of critical, so I'll just pause here for a second to check everyone is still with me.
Yeah, I guess I had a quick question. So because I'm not aware how many hours and times per 24-hour window flies sleep. So does anyone know? Was it just like one sleeping cycle after the training phase?
That is a good question. I have no idea how sleep cycles work in Drosophila. I know the specific conditions where they took the flies and they left them just in the tube for 24 hours with water before they did the testing. But yeah, I don't know what the typical dynamics of fly sleep is.
Yeah, because I assume in Drosophila, the consolidation also happens during sleep, right?
Yeah, I would imagine so, but I don't know. I mean, go for it, Ken.
Can you explain the volume stuff? So there's a relative MG volume and relative lumen volume, and they both go down?
Yeah, so this is to jump ahead to the discussion. Their theory as to what's going on is that there is an increase in the number of microglomeruli, but a relative decrease in these volumes. And that's happening because there are newly formed small microglomeruli. So as they go through in the discussion, their suspicion is that it's not that like all the microglomeruli that were there have shrunk. And to give you some specific numbers, the change is there were in the mock trained animals and in the GA trained animals, there's about 27. And in these new, in the CVA trained animals, there's 32. So their estimation is that what's happened is they've added about 5 new microglomeruli in the animals that have been trained on CVA, and that these 5 are probably smaller than the existing ones, which would mean that there's more microglomeruli, but because there's 27 old ones and 5 new ones that are small, the average size of them is going to shrink, and the average lumen volume of them is going to shrink. It would be nice if they had some way of tagging the new ones versus the old ones, and then they could actually demonstrate that cleanly.
Wouldn't that pop out if they just gave us absolute numbers instead of relative? Because this is giving us sort of the relative size or relative volume. But if they just said, oh, we see a much greater volume in total of these MG.
Yeah, I mean, if they just gave us the exact sizes for each of the MG, then you'd be able to do this. Presumably,
if you add them up, because if they're now more and the total volume is actually more, we should see that.
Yes. Yeah, I mean, that's what you'd expect, I guess, if you're seeing a greater number of microglomeruli. You'd also expect the total volume to be increased, but they don't provide that data. But that, yeah, I agree with you. That would probably happen as well. So we've established structural changes after learning that are specific to the odor and the projection neurons that are detecting and transmitting information about that odor is where we're at. One thing, and this is going to be very relevant to the end of this discussion when we talk about the potential experiments that we could design to decode memories based on this sort of work. But one thing I'm very curious about when I look at these data that are looking at relative microglomerulus volume and relative lumen volume and DA1 microglomerulus number is how correlated these changes are with each other. Is it the same flies? Because you can see the variation here is quite high in the CVA flies compared to the GA flies and the mock flies. And you can see a clean group difference, but it would be very hard to see things at the single fly level, as opposed to sometimes in like the mouse engram studies where you can see like these extremely dramatic between group differences with relatively small variance. I'm wondering whether it would be possible to do things at the single fly level if instead of just looking at one of these features, you looked at multiple of these features at once and had some sort of composite description of the changes. That's just something to keep in mind as we continue discussing this work. Oh yeah, and then they also had this control where they had flies that I think were knockouts from memory, although I should have said that. They were rut knockouts, so basically flies in which this calmodulin-dependent adenyl cyclase enzyme was missing. So this is an enzyme that is necessary for long-term memory formation in Drosophila, this sort of CAM-K equivalent enzyme. And indeed, they show that in these animals that are lacking this, this enzyme, these animals aren't able to learn to pair the odor with the particular sugar stimulus that they receive. And in these animals that can't learn, they also don't see any of these structural changes happening within the microglomeruli. So just another sort of negative control to confirm what they're seeing really is associated with learning. Okay, we're getting through the data. We've got one more key sort of interesting bits of data to go through. So this again in A here is examining the mushroom body calyx. And here they're labeling the postsynaptic Kenyan cells. So the dendrites of these Kenyan cells are being labeled in green. And the axon terminals of the DA1 projection neurons are being labeled in red. So that's the same as before. But this time they're also labeling the axons, like the full axons, not just the axon terminals of the projection neurons from the DA1 projection neurons in yellow using this modified YFP. And what they're going to use that to do is they're going to use that to trace through the mushroom body calyx of these animals and look at the distribution of the axons going into the calyx and where these axons are terminating within the calyx. Now, I, as a non-connectonicist, I saw this and I'm like, hold on, axon tracing using confocal microscopy. I thought the whole point of why we need electron microscopy is to do axon tracing because axons are normally very small. But I'm led to believe the reason why they can get away with this here is because they've only sparsely labeled the axons. And so even if they can't discriminate them very well, as long as they're sparsely labeled, they're still able to trace them using this sort of confocal microscopy technique. But if anyone wants to correct me on that, they're also welcome to pipe up about that at any point. So yeah, this is the starting point. They've got the termini, the synapses labeled. They've got the axons labeled. They want to look at the volumes. And what they find when they do this quantification of the volumes is first in F, they see that compared to if you just thought that these axon terminals were randomly distributed, it seems that the projection neuron terminal locations within the calyx is very much not randomly distributed. They sort of clump together into particular aggregates. And that happens for all the different ones. So whether it's animals that were trained to have positive associations with GA or with CVA or that weren't trained at all, these DA1 projection neuron axon terminals, they come pre-existingly clumped together rather than randomly distributed throughout the calyx. But what they do find happens due to learning is that the length of the axons increases in these CVA animals that are positively associated with sugar. And also the entire volume that is innervated by these axons increases in animals that had CVA as their CS+. So what they think is happening is that this is more evidence for the fact that the number of microglomeruli are increasing in these animals, and also the axon lengths are increasing in these animals. So their point is probably what is happening in these animals that have had CVA as a positive stimulus is that they're forming new microglomeruli and they're extending new axons to make contacts with these Kenyan cells and their dendrites as part of forming these greater number of contacts to consolidate this long-term memory. So this is the sort of key conceptual idea that the authors want to put forth in this paper. Just lastly, this is a little bit less relevant to our journal club. They also wanted to look at whether these sort of new contacts, these long-term memory consolidation changes, were actually associated with any changes in the Kenyan cell activity. So just to quickly run you through this, they have one final study where they look at the calcium dynamics in these Kenyan cells. So again, they have these flies that have calcium indicators in the Kenyan cells. So they're able to look at what is happening downstream of these DA1 projection neurons. And they show that in animals that have undergone the long-term memory training, so these are CVA, CS plus animals that have already undergone long-term memory, they show that when they are exposed to CVA, as opposed to just ethanol, which is the carrier solution, that you see a greater proportion of the mushroom body calyx seeming to show activity in response to the presence of CVA. So you can see the fraction of regions of interest that are responding to CVA increases pretty substantially in these animals that have been trained. And they also see that not only does the fraction of CVA responding regions of interest increase, but the dynamics increase a bit as well, where the offset of the dendritic firing, or dendritic activity rather, after the presentation of CVA becomes sharper. So basically, more of the mushroom body is responding, but it's going back to baseline quicker after the presentation of CVA. This isn't so important for the sort of structural changes associated with learning, but it's sort of just a cool piece of data to finish off their study. So just to summarize what I think this paper is showing us, it's that we see that these olfactory receptor neurons specific to a specific odor receptor are projecting to a specific stereotyped antennal lobe glomeruli, such as DA1. So CVA odor is always sending information through the olfactory receptor neurons into DA1, which has this stereotyped location within the antennal lobe. This antennal lobe contains projection neurons, which synapse onto a random set of Kenyan cells through approximately 30 microglomeruli or so. And if these animals undergo appetitive pairing of an odor and sugar, then what you see is increased microglomeruli, a decreased average microglomeruli volume, so presumably the addition of small new microglomeruli, and an increase in the innervated volume and axon length of these projection neurons into the calyx. And so the argument that the authors make is that long-term memory consolidation for appetitive odor memories likely happens, at least in part, through the formation of new microglomeruli in the calyx of the mushroom body. That's not to say that there isn't a whole bunch of stuff also happening downstream in the mushroom body proper, as per the traditional classical views of how learning and memory is happening within the mushroom body, but they are adding in what is happening in the calyx into this, showing that the calyx is not just a static feed-forward section, but is also doing things that are associated with learning and memory. Oh yeah, and then finally they show with their calcium imaging results that these structural changes are associated with altered Kenyan cell dynamics in response to the odor of interest. And then I see we have 15 minutes, so maybe I'll just run through this briefly and then we can open up to general discussion. But when I was reading this paper, it made me think of a very simple memory decoding experiment that I'm not sure would qualify for winning the memory decoding challenge, but at least I think puts good pressure on what should or should not count as a winner for the memory decoding prize, assuming that something like this would actually be feasible. But my thoughts were, what would happen if the authors did something like they repeated this specific experiment with very few modifications, but instead of trying to determine whether the long-term memory was occurring at flies at the group population levels, they somehow were able to do so at the single fly level. Now, I haven't read enough Drosophila behavioral papers yet to see whether this is maybe feasible or definitely not feasible or definitely is feasible. I know in this particular paper we were looking at, they were using a very short training period. So it was only a couple of minutes and a single occasion with the odor pairing with sugar. But you could have a spaced repetition cycle where you do multiple sessions of pairing the odor and the unconditioned stimulus. And maybe you could also do a better job of screening the fly behavior afterwards at an individual level to see whether you had a clean behavioral signature of whether an individual fly had actually learned the association or not. And then similarly, maybe you could also look at multiple features within the calyx or elsewhere. So not just microglomerulus size, or not just the projection neuron axon length, but a whole bunch of these at once to develop some sort of composite. Perhaps also using increased resolution instead of just this confocal imaging. And then if you could do all those things, perhaps you could give researchers some of these flies that had this robust sort of behavioral training, but you wouldn't tell them which odor they had, the flies had their conditioned stimulus to. So you wouldn't know whether these flies had come to like CVA, or they'd come to like GA, or they'd come to like the smell of bananas, or any of these particular odors. And then you ask them to determine which odor was the CS pairing only through examination of the mushroom body calyx by counting up the number of microglomeruli or other sorts of physiological features. Would that be enough to, let's say, decode one bit of information? Like, did these animals have a positive association with CVA or not? Now, there's immediately some sort of issues. Randall, I'll just get through this quickly and then... actually, no, I'll pause here. Yeah, go for it, Randall.
Maybe you could have finished first, but I guess I'm confused because we're stating here they should be doing this at the single fly level. But how exactly do they measure the increase in or decrease in volume, increase in number? I'm assuming they're doing this with confocal microscopy, looking at the calyx of a specific fly, and then they're doing it for a bunch of flies. So this must be done per fly, right?
No, you're correct. I have spoken badly, and this is a good point to get in with a clarification. The physiology is obviously all done at the single fly level. The behavior is not done at the single fly level. The behavior is done at the group level. So when they're measuring like the performance of learning in the flies, like how discriminatory they were for the conditioned stimulus, so CVA, as opposed to for the a neutral stimulus like GA. They literally, like, they've got the animals and they look at how many flies go into the CVA tube versus how many flies go into the GA tube. And, you know, at each individual fly, it may have just been randomly flying around between the two different tubes, but they're able to see at the group level
somehow to know which fly is which as they're going along. Okay. Yeah.
Yeah. And there are other studies which seem to do that a bit, but I just imagine it would be a lot cleaner if they knew that, like, the particular flies you were doing physiology on had learnt individually a very strong association or failed to learn a very strong association with the odors. Otherwise, it's going to be very messy in terms of your statistics later. So yeah, that's actually the next point I was going to make, is that there's likely, with this particular paradigm they use in this particular study, insufficient discriminatory power in terms of behavior at the single fly level. I mean, and this is why their physiology data might be fairly messy, because you're seeing this like high level of between group variation, as you can see on the right here. But that might be because there's a bunch of flies that like didn't actually learn the pairing, that are like messing up the data. And if you knew all the flies had really learnt the pairing or not learnt the pairing, maybe it would be cleaner. At least that's what I'd expect from mouse behavior, where I certainly am used to seeing that level of variation. And it's just because some of the mice didn't learn properly. And in paradigms where they do learn properly, it's a lot cleaner. But I'm less of an expert with Drosophila. Yeah, and I was just briefly looking at, there might be some paradigms where you're able to have a cleaner view of what's happening at the individual fly level. But then the other sort of critical thing is, even if you did do this and you succeeded, this would only be like one bit of information, which is, I mean, it would be a very cool experiment, but it's arguably still trivial if you can only get one bit of information. I'm curious as to what people's thoughts are on that, and what would be the case if you could do more than one odor of discrimination at once. Maybe you could do which odor did they come to like out of not just CVA or not, but it would be CVA or GA or other smells. But that does get more complicated. And it depends on how cleanly the different odors can be distinguished and mapped onto these different projection neurons and the glomeruli from which they originate. Okay, we have 7 minutes left, so it's now very much open to general discussion, starting with Ken, but then anyone else can jump in at any time. Go for it, Ken.
So the experiment that they did here was appetitive because they paired it with sugar. Is there any like experiment that you came across or in this paper where it was an aversive stimulus? Because I assume that I assume that this is a gain control. That this has nothing to do with kind of learning. It's saying, hey, this is an important odor, and we need to, whether it's good or bad odor, it's an important odor, and it needs to be better represented in the Kenyan cells.
Yeah, in the discussion, I think they talk about how they don't have the aversive data, but they suspect the same thing may happen if you used an aversive stimulus rather than just an appetitive stimulus. I'm not aware of an aversive stimulus equivalent of this particular paradigm. And I agree with you that it may well be that it's, you know, what the mushroom body calyx is doing is gain.
Is gain, valence, or importance not a type of memory?
Well, so that's the point I was going to get to, where it still is like, you know, it's a change in behavior. It's a change in structure that results in a change in behavior. It's just like how specific is it would be an issue.
If I can just go off for a second here. So I don't know how many different types of odor receptors there are. I think 50, 60 type of. Yeah,
that's right.
Okay. So the number of these types of experiments that are happening at the level of an individual odor receptor is far less than the number of individual odors that could be discriminated in the thousands of Kenyan cells that make a combinatorial code off of them. And so there's an argument to be made that there really is a high fidelity memory system, but it's farther downstream. And the only reason why this is, you know, a classic case of they could not label in this experiment a set of Kenyan cells, although I feel like, you know, the calcium imaging could do that. But they had a system that they could get their hooks into, and they said, okay, let's go ahead and do it, even though this is kind of a less interesting part of a much more interesting system that encodes memory.
Yeah, I agree with all of that. I guess my question, though, is like, is this still enough to count? So let's say they were able to do a few different odors. So like not just CVA, but they could do 3 or 4 different odors at once. And they were able to see that, like, you know, it really was the specific microglomeruli associated with that specific odor that they were able to pick out reliably. So they could trace it from DA1 or from a different set of projection neurons. And they're able to say, yeah, look, we see it goes from like 20 microglomeruli to 40 microglomeruli reliably. And we know the projection neurons they come from. So we know the smell that animal had, even if it's just that the gain has gone up for that particular smell, for that particular fly. Would we count that, or would we say that's not good enough? What do you reckon, Ken?
My gut instinct is that it would not be good enough, but I don't know what that, you know, that's just my input. I could argue that, so if I had a dog that I left out in the winter, and I measured its fur, and the fur was longer, and that was correlated with how cold the winter was, is that a memory? I know that's a crude thing, but it's another. Gain control is a very crude thing to call a memory. Although I realize that it's not a clean definition.
Yes, I don't know how, I can't read Cyrillic. So
yeah, my bad. I had to reconnect from another account. My name is Arseni. I feel like I've heard that, and I go by this definition, so I'm curious what you guys think about it, is that any trace left on CNS can be labeled as memory. So in terms of the fur, well, it's the brain controlling the controlling what's going on with the body, right? So
yeah, I mean, this is what we discuss a lot. There are some things that are sort of trivial. As a dramatic example, I can't remember whether it was Conrad Cording or you, Randall, or someone else who used this example. But like, if you hit someone in the head and you cause brain damage, you're going to have like very clear signature of behavioral changes afterwards associated with a very clear structural change. But we tend to think of that as sort of like a trivial change where like there's not a clean, interesting structural change associated with the behavioral change. What we're going for with this memory decoding challenge is like a sort of, when we say non-trivial, we mean that like it's encoding information in an interesting way that's associated with what the animal does ethologically, where it's sort of like multiple bits of information at least. Yeah, this is kind of the philosophy that we're getting into. You could say that any of these things are memory, but it's how cool is the memory.
What this discussion points out is that it seems like we're maybe adding an extra bit to the requirements of the challenge, which is kind of, you have to be able to pick up on some interesting non-trivial amount of information within a single animal. Because if you were using this instead to just categorize all your flies into these are the ones that have been trained for that, and these are the ones that have been trained for that, you could say, okay, you're doing something useful here and you've got multiple bits because you're categorizing your whole group of flies into different sets based on what you're measuring. But yeah, it's not inside a single fly. Yeah.
Or I was thinking another, leaving the dog out or bashing someone in the head. I was thinking more like tree rings, right? You can tell a lot about what a tree has gone through in the past through the tree rings, but I wouldn't really consider that a memory of some kind, even though you can tell the past objectively by looking at the structure. I'm skeptical. Well, let's just say I think there's a lot more work to be done in this.
I love that example of tree rings, because that not only is it an example, but it has many, many bits of information that can be easily read out. I'm going to steal that one if that's okay.
Nice.
Yeah, go ahead.
So we're close to the top of the hour. Does anyone else, particularly if you haven't spoken, have any questions or comments before we finish up?
All I want to add is that this is, I think, the first time that I've been flung headfirst into trying to actually learn about what's going on in fruit flies, because previously it's been mainly a matter of looking at how good or how bad the various EM data was and tracing and those kinds of technical things. And then eventually learning that, oh, the cells look different. They've got this soma that hangs off to the side, and you have to look at them differently than when I look at mammalian cells. It's the first time I've really looked into, I guess, circuitry of fruit fly. So awesome.
Yeah.
Thanks.
That was a good start. I think also simpler than trying to get into all those different modules within the mushroom body initially. So we can do another one next on actually all the different lobes, the alpha, beta, alpha prime, beta prime, the dopaminergic modulation, everything, and see if we've got another experiment there, which can be clarificatory. But I enjoyed this, and I hope you guys did too. So any last questions just to check before we finish up? Otherwise, we will see you all at the next one in 2 weeks from now.
Thank you, Ariel.
Cool. All right. See you all next time. Thank you very much for coming.
See you guys.
Bye-bye.
Bye.